A flower emerges from frozen ground in late February, pushing through snow when no other plant dares to grow. Authorities analyzed this botanical anomaly for decades before making an unprecedented decision in 1984. They officially recognized it as medicine, listing it in Commission E monographs alongside pharmaceuticals. The plant demonstrated such consistent results in clinical trials that it earned legal medical status across Europe. Then, in the early 2000s, regulatory agencies suddenly reversed course. They declared the same flower toxic, recommending against its use despite thousands of years of documented safety. The timing was suspicious. Pharmaceutical companies had just developed synthetic alternatives to natural expectorants. Patents worth billions depended on eliminating the competition. This flower offered something the industry could never control. It grew wild, it reproduced freely, and it worked without modification. The plant is Coltsfoot, known scientifically as Tussilago farfara, and its story reveals the uncomfortable truth about modern medicine's relationship with nature. If you enjoy learning about plants that challenge the pharmaceutical narrative, subscribe to this channel for more forgotten botanical knowledge. Coltsfoot blooms in temperatures that would kill most flowering plants. It emerges when the ground is still frozen, sending up bright yellow flowers on scaly stems before a single leaf appears. This reverse growth pattern confused early botanists. The Latin name Tussilago means cough dispeller, referencing its primary traditional use. The genus demonstrates remarkable cold tolerance, surviving in zones as harsh as 40 degrees. After flowering, large hoof-shaped leaves emerge, giving the plant its English name. These leaves have a distinctive dual texture, smooth green on top, white and woolly underneath. The plant spreads aggressively through underground rhizomes, colonizing disturbed soil and riverbanks. It thrives in clay-heavy, mineral-rich environments that drain poorly. This ecological strategy makes it a pioneer species, stabilizing erosion-prone areas. Indigenous peoples across Europe and Asia recognize these leaves immediately. They collected them in late spring when the woolly underside was most developed. The timing mattered. Chemical composition shifts as seasons progress. Ancient herbalists understood cycles that modern science would later confirm through spectroscopy. Traditional medicine systems from China to Greece documented identical uses for coltsfoot. Historical texts describe preparations for bronchial complaints, asthma, and persistent coughs. Medieval monks cultivated it in monastery gardens specifically for respiratory ailments. The active compounds include mucilage, which forms a protective coating over irritated throat tissue. Tannins provide astringent properties that reduce inflammation in airways. Flavonoids offer antioxidant effects that support healing in damaged lung tissue. German researchers isolated these compounds in controlled studies throughout the 1970s and 1980s. Commission E, Germany's equivalent to the FDA, reviewed extensive clinical data. They concluded the evidence supported traditional uses for coughs and bronchitis. The approval came with specific guidelines, external preparations, and historical documentation only. For decades, European apothecaries sold standardized extracts. Then, laboratory analysis detected pyrrolizidine alkaloids in the plant. These compounds, present in trace amounts, can cause liver damage in extremely high doses. The discovery triggered a regulatory panic. Authorities banned or severely restricted coltsfoot across multiple countries. The pyrrolizidine alkaloid controversy reveals a pattern in botanical regulation. Natural substances cannot be patented unless chemically modified. Pharmaceutical companies invest billions in synthetic drug development. A wild plant that grows freely represents zero profit potential. Coltsfoot demonstrated clinical efficacy comparable to commercial expectorants. It cost nothing to cultivate and required no prescription. The alkaloid content provided the perfect regulatory mechanism. Authorities cited potential liver toxicity, despite the absence of documented cases from traditional use. Historical consumption spanning millennia showed no epidemic of liver disease. The dosage required to cause harm far exceeded anything in traditional preparation methods. Yet the regulatory response treated the plant as uniformly dangerous. Meanwhile, synthetic cough suppressants with documented side effects remained widely available. The distinction was not toxicological. It was economic. One substance could be controlled, prescribed, and monetized. The other grew in ditches and belonged to no one. Plants produce pyrrolizidine alkaloids as natural pesticides. These compounds deter herbivores and protect against fungal infection. Coltsfoot contains low concentrations compared to other PA-bearing species. Concentration varies dramatically based on growing conditions and plant part. Flowers contain negligible amounts. Leaves harvested in spring have lower levels than those collected in autumn. Modern analytical chemistry can measure these compounds in parts per billion. Studies show traditional preparation methods reduce alkaloid content significantly. Water extraction pulls primarily mucilage and flavonoids, leaving alkaloids behind. The plant developed these defenses over millions of years of evolution. Human liver enzymes can process small amounts without damage. The problem emerges only with prolonged exposure to concentrated extracts. Traditional use involved short-term applications during acute illness. No herbalist in recorded history recommended continuous daily consumption. The wisdom was embedded in practice: use when needed, stop when symptoms resolve. Modern regulation ignored this nuance entirely. Coltsfoot originated in Europe and northern Asia. Roman soldiers carried it across the empire as standard medical supply. It naturalized in North America following European colonization. The plant now grows wild across temperate regions worldwide. It prefers disturbed ground, construction sites, roadsides, gravel pits. This ecological role makes it valuable for land reclamation. The extensive root system prevents soil erosion on steep banks. Miners in Appalachia recognized it as an indicator of mineral-rich soil. Gold prospectors in California found it growing near promising claims. The plant's presence signals specific geological conditions. It tolerates heavy metal contamination better than most flowering species. This resilience made it useful for phytoremediation in industrial areas. Botanical gardens worldwide maintain collections for research. The Royal Botanic Gardens at Kew hold specimens dating to the 1700s. These preserved samples allow modern scientists to study historical chemical composition. Analysis reveals cultivated varieties once existed with lower alkaloid content. Agricultural selection could have produced safer medicinal strains. That research stopped when regulatory bans made cultivation pointless. Different nations responded to the alkaloid data with contrasting policies. Germany maintained restricted approval for external preparations. The United Kingdom banned sale for internal use but permitted cultivation. Canada prohibited all medicinal claims while allowing ornamental sale. The United States took no federal position, leaving regulation to individual states. Traditional Chinese medicine continued using the related species Tussilago farfara. Japanese Kampo medicine retained it in classical formulations. The cultural divide reflected different relationships with plant medicine. Western regulatory systems demanded zero-risk guarantees. Eastern medical traditions accepted risk-benefit calculations. The pharmaceutical model treats all substances as potential poisons requiring proof of safety. The herbalist model treats plants as complex organisms requiring wisdom in application. Neither approach is absolutely correct. But the regulatory shift eliminated consumer choice entirely. No label warning, no dosage restriction, no time limit guideline. Just complete prohibition based on theoretical rather than documented harm. Coltsfoot still blooms every February in disturbed soil across continents. The bright yellow flowers emerge from snow, indifferent to human regulatory debates. Wild populations thrive in industrial zones and abandoned lots. The plant requires no cultivation, no irrigation, no fertilizer. It survives because it adapted to harsh conditions long before humans existed. The leaves still develop their distinctive woolly texture. The mucilage still forms when fresh material contacts water. The chemistry remains unchanged despite regulatory reclassification. Knowledge of the plant persists in herbalist communities. They preserve historical texts documenting traditional applications. They understand the difference between documentation and recommendation. The plant itself makes no medical claims. It simply grows, reproduces, and continues the biological strategies that ensured its survival. Whether humans choose to study it, regulate it, or ignore it makes no difference to the species. Coltsfoot will outlive the patents, the prohibitions, and the pharmaceutical companies. Nature operates on timescales that make human institutions look like temporary experiments.